everyone, welcome to my channel. And today I come with another topic. This is coenzyme, and you just see the my previous videos about the enzymes, that is related enzyme, actually double reciprocal plot and inhibition, enzyme inhibition, and the Michaelis-Menten equation. Everything I described in my previous videos. You just check out that videos, and after that you follow this one. Okay, so today's topic is that the coenzyme. What is coenzyme? We know about enzymes, but enzymes is not the coenzyme. So what is the coenzyme? Actually, many enzymes that has the two parts. One is the protein part and another is the non-protein part. Clear? One part is the protein part, another part is the non-protein part. Okay, so generally the protein part, this is called the apoenzyme. Okay, you just learn what is this coenzyme, and after that we learn about the activity. What's the role of the coenzyme? Okay, so actually any enzyme. Suppose this is enzyme, it has a two part. Clear? This is the proteinaceous part and this is the non-proteinaceous part. The proteinaceous part, the protein part, this is called the apoenzyme. And which one is the coenzyme? The non-protein part, this is called the coenzyme. Clear? And totally apoenzyme and coenzyme both all together is called the holoenzyme. Okay? This is called the holoenzyme. And there is a main role of the coenzyme, that is when the coenzyme actually bind with the apoenzyme either non-covalently bond or with the help of covalently bond. Okay, so in case of when the coenzyme that binds with the apoenzyme or linked by the non-covalent bond, then it gets easily detached after the enzyme activity. Okay, after the catalyzation activity, we know that enzyme generally increase or catalyze the reaction rate. So after the activity or after the reaction, the part coenzyme part, this is totally detached from the enzyme part. But sometimes when the coenzyme that linked with the apoenzyme with covalently bond, this happens when it is detached. This happens in case of non-covalent bond. Clear? But in case of covalent bond, when the coenzyme bind or linked with the apoenzyme with the help of covalent bond, then we call the coenzyme is the prosthetic group. Clear? This is called the prosthetic group. And here, after the reaction or after the catalytic reaction, it does not detach from the enzyme. It actually remains with the enzyme. This is the difference between the coenzyme actually arranged or it linked with the apoenzyme. When it binds with the apoenzyme with the help of non-covalent bond, then it is detached. But when it binds with the covalent bond, they cannot detach from the enzyme and it remains attached with that. In that case, it is called the prosthetic group. And here, two examples are there. That is, one is NAD plus and another is the FAD. Here, NAD plus is the example where it is detached from the lactate. Suppose this is one kind of enzyme known as the lactate dehydrogenase. So, when the reaction is over, the NAD plus totally detached from the lactate dehydrogenase. But this is another example, FAD. This is actually we see in the succinate dehydrogenase, and this is called the prosthetic group because it cannot detach from the succinate dehydrogenase after the catalytic activity or after the enzymatic reaction. Okay, so we learned here about the coenzyme. Now, what is the role of the coenzyme or the activity of the coenzyme? This is here. So, what is the role? It generally helps in binding of the substrate to the active side of the enzyme. We know that enzyme has the active side, that is generally for the substrate binding site. Clear? So, what is the role of the coenzyme? It generally helps for the binding of substrate with the active side of the enzyme. Second point, it accepts the atom or the any functional group, or it removes the electron from the substrate. Okay, it is another role of the coenzyme. That means it helps either it accepts any functional group or atom, or it can remove the electrons from the substrate. Clear? So here, this is the reaction. You see, this is an example. Suppose this is the substrate, and here the hydrogen is bound with the substrate, and NAD plus here is the coenzyme. What happened? And this is the enzyme dehydrogenase. Okay. Suppose NAD plus here this is the part of the dehydrogenase enzyme. So what happened? We see that AH2, when transformed or oxidized form of A, or the hydrogen is released from this A, it is totally attached with the coenzyme to form the NADH. Clear? And that means it just remove the hydrogen atom. Okay, NAD plus that remove the hydrogen atom from the substrate to help or oxidize the substrate. Okay, and it actually form another compound. This is called the NAD. In the second step, you see that NADH and H plus in presence of another dehydrogenase. This is known as the NADH dehydrogenase, or the mitochondrial NADH dehydrogenase. And here what happened? NADH that is released. Okay, that is released and to form NAD plus. That means the hydrogen atom which is already attached with the NAD. Clear? This totally separated from the NAD and to form hydrogen, and NAD plus again form. Clear? So first of all, you see here how it removes the hydrogen, and this is how the compound that containing hydrogen, it removes its hydrogen to form again NAD plus. Clear? And third point, it actually donates the group or the atom to be added to the substrate by enzyme. Clear? Here we see that it removes or accepts. Here we see just that it donates the group or atom. Clear? It donates the group or atom to be added to the substrate. This is an example you see, and it's known as the tetrahydrobiopterin. Clear? And here hydrogen is already attached with this one. Clear? And when it reacts with the tryptophan oxidation process occurring, and tryptophan that form 5-hydroxytryptophan, the tryptophan that transforms to 5-hydroxytryptophan, and the tetrahydrobiopterin that convert into the dihydrobiopterin. Clear? So these are the activity, and here this is the coenzyme. What is coenzyme? Clear? The definition and their types, and this is the activity or the role of the coenzyme. Clear? So this is the coenzyme. Now we learn about the allosteric protein, isozyme and the ribozyme. Okay? Now we learn about the isozyme and the ribozyme. What is isozyme? Isozyme, and it is an enzyme that occurs in different molecular forms within the same species. Okay, if there is any species that contain different molecular forms of enzyme, okay, one enzyme but that remain in different molecular form. Okay, enzyme is one, but we can see it in different forms, and it present in the same species. Okay, it present in the same species, and such different molecular forms of the same enzyme are called the isozyme. That means here what we learn here, that isozyme is nothing but one enzyme that remain in the same species but in the different forms. Okay, so here the differences are what? That means the molecular forms are different. Enzyme has the different molecular form, but what difference another that separate that all the enzymes from one to each other. Okay, that means amino acid composition is not same all the form. Number two, the electrophoretic mobility is not same. Okay, these are the you think these are the another type of you sometimes you cannot understand. You can see about this in biochemistry, that is Harper's book you follow about. So you learn these terms, about these terms, and if I will make any videos about the protein structure or the carbohydrate structure or the lipid structure, then I will explain about these terms. But today the topic is isozyme, so we have to focus this part, okay? And if you have any problem, then you can see these terms that are present in biochemistry book, like Harvard's, you can follow, okay? So these are the and styles also. So first one is the amino acid composition. It is not same in the all forms. Electrophoretic mobility is not same. The thermolability is not same. Immunological properties must be different, and allosteric properties also different. Here this is one kind of enzyme, that is the lactate dehydrogenase, and it is discovered that it remains in 5 different forms and in different vertebrates. Okay, so this is the isozyme. What we learned, that one enzyme but remain in the different forms. And this is the example, lactate dehydrogenase, and we see this lactate dehydrogenase in 5 different forms. Now we learn about the ribozyme. What is the ribozyme? Ribozyme actually it is sometimes enzymes that are, generally we know the enzymes are the proteinaceous in nature. So sometimes some RNAs, they have the some activity like that, they can play the role about like that enzyme. Okay, ribozyme means the ribosomes that play the role of enzymes. Okay, that means what is written here, enzymes are the proteinaceous in nature, and few RNA within the ability of the catalyzing certain biological reactions. Okay, it helps in the catalyzing reactions, but it only helps in the biological reactions. And these are the ribozymes. Here you see, this is the structure, and first you see this is the E. coli tRNA, and after processing this form the mature tRNA. Okay, and you just follow this part. This is the 3' part and this is the 5' part, and this part is known as the 14kD protein. Okay, 14kD protein, and this part is the extra sequence. Okay, and this is also the extra sequence, and both actually totally call, or both together we call the RNA-seq. Okay, RNA-seq. Sorry, 1 second. Sorry for the disturbance. Okay, so what we learned about that ribozyme? Ribozyme is the RNA. When RNA act like the enzyme, or it has the ability of the catalyzing activity sometimes in the biological reactions. So here you see this is one kind of the E. coli tRNA, and that contains 14kD protein and the extra sequence. Here is one extra sequence and another is, this is the extra sequence, and totally this part is called the RNA-seq, and 14kD protein, this is called actually the ribozyme. When the E. coli tRNA that converts into the mature tRNA, you see there is no extra sequences. Okay, the 3' extra sequence that is cut off by the exonuclease, and the 5' is cut off by the RNA-seq. Okay, the 5' is cut off by the RNA-seq, and it is discovered, or it is found that if the RNA-seq or the 14kD protein is not present, then these extra sequences that are not cut off. Okay, that are not cut off in in the processing of the mature tRNA. So this is the ribozyme and this is the isozyme. Okay, in my next video I will discuss about the allosteric protein. What is allosteric regulation? Okay.